So briefly, my outline, I'm going to talk about our research question, the approach that we've taken technically. I'll show you some example images, hopefully to pique your interest a bit as I get through the two slides of text on results. And then a bit about the challenges that we've experienced in developing the equipment and in using it on routine surveys. Some examples of the data products that we're able to provide. And then finally, of conclusions and some of the relevance that I see to the session theme. So our basic research question is, can we accomplish the principal sampling tasks of a fishery survey underwater? These being count by species and getting individual lengths of these species. Some thoughts on why we'd want to do this underwater. We can improve existing surveys. Importantly, get more information with the same sampling effort. We can preserve information on three-dimensional distribution at very, very fine scale. We can look at patchiness, species and size overlap. We can reduce the problem of selectivity in the cod end. With this gear, we're able to sample individuals from cod over 1 meter long to euphausids, at sub-centimeter scales. We also don't discard our samples when we discard the catch because we have a full inventory of all of the images. So it's possible to, in fact, reuse that survey data in completely new ways at later dates. Also, things like quality checking catch information. And then I have a couple of thoughts on new techniques potentially for future surveys related to doing trawling with an open cod end. Where we would have reduced mortality. Fish are simply released at the same depth that they were captured. We could do long transect surveys, sort of thinking about the mega transects that are used in terrestrial ecology. You don't have to worry about your cod end filling. And also trawling at much higher speeds where you don't have the towing resistance of a closed cod end. So the way that we're using the system, we're placing the camera system in the aft portion of the trawl between the extension and cod end. And the reason is problem of scale, that we've operated primarily in pelagic trawls where we're looking at tens of meters scale openings. And we really have an effective range in the somewhere around 3 meters with imaging technology. So if we move the system back into the extension, we're in 1 to 2 meter opening, and we can get an image of everything passing through the trawl. So here's an image of the system. This was from a cruise this summer. On the left side is leading back into the cod end. To the right, it would be up towards the opening of the trawl. And this is an image taken underwater with a towed vehicle just over the system. Again, the cod end to the left and the trawl opening to the right. So a little bit about the technical aspects. The main camera unit is less than 1 meter long. It weighs 26 kilograms in air and about 3 kilos in water. The image on the right is how it's placed in the larger deployment frame in the trawl. We have stereo color cameras, 1.4 megapixels, and we collect images at 5 frames per second, everything time referenced. And also inside this, the main unit is a PC to control the cameras. It's saving the images. And we also have capacity to have a live feed to the vessel. Light is provided by external LED strobes. They give 30,000 lumens of light. That's equivalent to about 35 incandescent 65-watt bulbs. So it is a lot of light that we're introducing, which I know can be of concern, certainly related to fish behavior. 2,000 meter depth rating, and we're running it off a battery and can get 8 hours of runtime. So some example images. If you can click on this one, this is actually a video. This is running at 5 frames per second. So this is exactly what the data collection looks like. If you were looking at it on the bridge, this is what you would see. Here I've compressed 4 hours of trawling into 31 seconds by removing the time without any fish. So the range over which we were sampling was 15 to 131 meters, but we know the precise depth at which each of these objects passed through. So a bit about the results. So we've looked at verifying, well, are we getting the same results from the images as we do with standard sampling of the cod end? So this is an example of 9 hauls. We had 16 species of fish captured and 1,548 individuals. You can see we were 18 short in our images. And this was a problem with the deployment. There was a small area that was outside of the field of view. So I think a few fish went past. That's something that we've remedied. But overall, our error is 1% in total count. And then we're also able to measure lengths. And we've tested this in 2 ways. The first, we had 5 fish in a pen and just let them freely swim back and forth in front of the camera over 10 hours. So we know the length of each of these fish. And here we're able to compare the mean length calculated from the images against the actual length of the fish. You can see for all individuals we were within 2.3% for the length. Now when we're working actually on a survey and in a trawl, we can't link an individual fish with an individual image because everything is mixed in the cod end. So here the approach we've taken is comparing length frequency distributions. So in these plots, the hashed lines are what was physically measured from the cod end catch, and the solid line is the length frequency distribution calculated from the images. So you can see we get very good fits of the sampled population. A bit about the challenges. First question that anyone asks is, well, what happens when the water is not clear and you're relying on images? It's actually not as big of a problem as we thought it would be. This is this example image is from a monochrome camera, which we had direct lighting. Yeah, so that was not a video. I don't know if you can make it out, but that blob in the lower right-hand corner is in fact a cod, which I know because 10 seconds later it passed through our system. The major difference here is that we're using indirect lighting. So I think we're not illuminating the particles in the water nearly as much, so we're still able to get very usable images. Certainly a little bit diminished quality, but we can tell that that's an Atlantic cod, and I can do a length measurement on it. And we have, in fact, used the system in bottom trawl, and have gotten good images as well. So another challenge, the behavior of fish. We can get very high entrance rates. This is a video, if you could click on it. So here we had about 172 blue whiting passing per second. But, I mean, it still is possible to count the number of individuals with probably some small margin of error. And there are a large percentage of those individuals where I can see both the snout and tail and calculate a length. Another problem we have is fish that enter the field of view multiple times. You click on this one. So here we had a saithe who enjoyed the limelight and for 3 minutes and 15 seconds kept swimming back and forth in front of the camera. This is something that we also probably could remedy by putting a longer extension to the cod end. Or again, if we use the system with an open cod end, this fish most likely would simply pass out. So now the parts that I think are a bit more exciting, what are we able to actually produce? So here is a haddock who doesn't look very healthy. Appears to have evidence of barotrauma. You can see that the eye is distended. There's also, looks like bleeding inside of the eye. There's scale injury. But if we look, the gray line is the trawl profile, and we haven't brought this fish up to the surface. But there was considerable commercial fishing activity at the same time. So we think we probably encountered a fish that was discarded from the commercial trawl fishery. The other thing is that was the only haddock we found. So what was this haddock doing up in the water column when we know that the commercial fleet was catching haddock at the seabed? And then another ill haddock has a very large abscess in its lower mandible. And it's obviously a very malnourished fish if you look at the stomach fullness. The only thing that its stomach contained was krill, and the small flecks that are around it are in fact krill. So it was present in this significant krill layer. We can also look at small objects. I mean, here we're looking at zero group young of the year fish. So going from tens of centimeter and meter scale, we can look at single centimeter scale as well. Upper left-hand corner is in fact a Greenland halibut, which is nearly translucent, but we're still able to get good enough images to be able to identify and do length measurements. And we're not limited to fish. I mean, we can also look at things like jellyfish, small crustaceans, krill, amphipods, shrimp. Yep. And then this, I think, is the most exciting of the data products, where we can do a profile of what came through the trawl during the entire haul. So the top is the sea surface. Gray line, again, is the trawl going down and coming back up. Black line is seabed. So this was going across the continental shelf break in the Norwegian Sea. So we have the time and depth that each fish passed. We can look at species overlap. So you can see that the first species that we encountered were Atlantic mackerel with a few haddock mixed in. Then we went through a layer, it appears, of saithe, Atlantic pearl sides, and then this deeper layer of blue whiting with redfish mixed in. We can see the passage rates. Here I've scaled the markers to actually show the density of fish passing in numbers per second. And this is the part where I think it gets very interesting. We can look at the acoustic record. And so if I go back again, you can see there's that very distinct layer of blue whiting, but there's also some scattering a bit higher in the water column that looks like probably was the saithe. So we can really verify the species seen acoustically. And we can measure length all along the way for target strength analysis. For instance, we were in this blue whiting layer for about an hour. Well, we can look at if there were trends in fish size throughout that to be able to better correct for biomass estimates for the acoustic data. So just quickly, conclusions and what I see as the relevance to this session theme. We can get a full image record of the entire haul that's technically feasible. Our measurements of length match very well with manual sampling. So we're getting some very new products. And then some questions that I have for the group are sort of what are the survey and assessment needs that this data might fill, and how might we adapt or design new surveys to utilize this information. And then just acknowledgements. This is work done as part of my PhD, sponsored by Scantrol, then the Institute of Marine Research, particularly the CRISP Center. I want to thank the program leaders and cruise leaders for inviting me on cruises. I think I'm not taking an extra 10% of time, but and then the Research Council of Norway through the SFI Industrial PhD Scheme, and the University of Bergen.